The speaker for the second talk is Professor Siddhesh Kamat from the Indian Institute of Science Education and Research, IISER, Pune. Professor Kamat is currently an associate professor, a Swarna Jayanti fellow, and an EMBO young investigator in the Department of Biology at IISER, Pune. Professor Kamat completed his BTech from the Institute of Chemical Technology, which was formerly called UDCT, in 2007, and a PhD from the Department of Chemistry at Texas A&M University in 2012. Professor Kamat was the 9th Irving S. Siegel Postdoctoral Fellow of the American Chemical Society at the Department of Chemical Physiology, the Scripps Research Institute. He joined IISER Pune in 2016 and is the chairperson of the National Facility for Gene Function in the Health and Disease at IISER Pune. He is a fellow of the Royal Society of Chemistry. And this is the program sponsored by the Infosys Foundation, and Professor Kamat received the Infosys Prize in Life Sciences this year in 2024. So welcome Professor Kamat to the stage. He will give the talk on the board. If you can't see or something, if you need him to write bigger, please interrupt right at the beginning and tell him that. And what I forgot to tell last time, please put your mobile phones in silent mode. Thank you. Yeah, there's a column. Caller mic.
Am I audible? At the back? No? Better.
Is it on?
Now?
Is it on?
Still no? Is it on? Am I audible? Too audible. Better now? Okay. So I was told by Mayuri that I need to talk to students who are just sort of getting out of 12th grade or first year students. So I thought instead of speaking something specific about my research, I will generally talk about a subject I'm very passionate about, and that is enzymes. We work with enzymes a lot. Now, as you heard from my introduction, I have sort of trained as an engineer first, then a chemist, and now I'm in a biology department. So you can see how confused I am. So what we do is we use principles of engineering, we use principles of chemistry, we use principles of biology to solve problems associated with enzymes. A general advice to everybody, all the students in particular here, is please feel free to interrupt me anytime during my talk. If something is not clear to you, it's probably not clear to a lot of people. So you're doing a general service by asking me a question, so don't feel shy, however stupid you think your question is. Fine? So what I will do is I will sort of start with very simple things, and hopefully I will try to build up. Now, enzymes, heard of enzymes? Everybody? Good. Makes my life easy. What are enzymes? Enzymes are proteins. Enzymes are catalysts. Enzymes speed up rates of reactions. Okay? So let's start off looking at something which I'm sure most of you all have seen. This is an energy profile diagram where I'm looking at the energy on the x-axis, and I'm looking at maybe the reaction progress or the rates of reactions as I go along. Now, for a standard reaction where I'm looking at a reaction going from A to B, I have the energy of A, I have the energy of B, and I have an energy barrier to cross. All right? Seen this in textbooks? Good. Now, This state of the reaction is called? Excellent. Now a transition state of a reaction is the highest energy state. It is the only point in the reaction where you cannot isolate that intermediate. It is the most unstable part of the reaction. Okay? And this particular reaction, if I draw a dotted line along A, has two components. It has two energy components. Okay? It has energy of activation, which is represented by delta G, double dagger, and it has the energy of the reaction, delta G. Okay? Now this energy of activation is what is going to determine how fast the reaction goes. Alright, with me so far? Cool. Now what do catalysts do to this reaction? Okay, let's talk generally about catalysts. Catalysts have the following property. Number one, they don't transform themselves. They remain as such at the end of reactions. Number two, they are going to make reactions go faster, and if this reaction were an equilibrium reaction, catalysts would have no effect on the overall equilibrium of the reaction. It would only change the rate to reach equilibrium. So what catalysts do in this case is the following. They are going to typically reduce the rates of reactions by reducing the energy of activation. They're going to do something to the transition state. Interestingly, most of the times they're going to stabilize the transition state and therefore make the reactions go faster. Okay? So this is generically about catalysts. Now when I am looking at enzymes, there are a few more things I need to consider. Okay? So this is everybody with me so far? Alright. Now let's look at enzymes. You can read what I'm writing, right? At the back. So enzymes do the following things. So the nomenclature for enzymes slightly changes. The reactants in chemistry are now called substrates, and the products are still called products. They are denoted by the letters S and P. Okay? And again, most enzymes are denoted by E, and they catalyze in biology most of the times reversible reactions. Okay? Now there are several things happening in this reaction. So I'm going to slightly expand this equation. Alright? So you have a substrate, and you have the substrate bind to an enzyme which is a catalyst. Now this substrate and enzyme together form a stable complex which is called the enzyme substrate complex. So we can sort of classify this step as binding. Okay? Now when a substrate binds, it has two options. It can either dissociate and go back to a free substrate because it is not bound in the correct conformation on the catalyst, which is the enzyme, or the enzyme can catalyze its conversion to product. Okay? And we will call this part of the reaction as chemistry, or the chemical transformation. Alright? And now once the product has formed, it has two options. It can go back to substrate if it's a reversible reaction, or it can dissociate from the enzyme to give you enzyme and free product. So it will give free enzyme and free product. And this step is called dissociation. So you have three components in an enzyme catalyzed reaction. And about 99% of the cases in biology, the chemistry is the rate limiting step of the reaction. All right? So now let's try and redraw this energy profile diagram. So you have a step where I have the formation of maybe an enzyme substrate complex. I have chemistry, which is the rate determining step to give you my enzyme product complex. And then I have the formation of a enzyme and free product. Okay, so you can see how an enzyme, by reducing the overall energy of activation, is making reactions go faster. With me? Any questions? Okay, so depending on the step which is going to be the rate limiting step, one can decide how to draw energy profile diagrams for such reactions. All right, and we will look at two examples of how enzymes enhance rates of reactions. Okay, just to make you appreciate what enzymes do in biological timescales. So the first reaction is of an enzyme. So the good thing about enzymes is if you know what the name of the enzyme is, almost always you can predict what chemistry it is catalyzing. Okay? So the enzyme we are looking at is an enzyme called OMP decarboxylase. All right? And what this enzyme does is the following. It takes a this molecule, which is called orotidine monophosphate. I am not drawing the sugar part, but this is a precursor to making one of the essential nucleobases, which is uridine monophosphate. Okay, so that's the reaction it's catalyzing, and it is basically releasing CO2 in the process. Okay, so very simple decarboxylation reaction. Now the person who studied this is a guy named Richard Wolfenden. He's at the University of North Carolina, and he started trying to figure out how much rate enhancements do enzymes give versus uncatalyzed reactions. So what a student in his lab, who was a PhD student, did is tried to find out the rate of this uncatalyzed reaction. So what he did is he took this compound, he kept this at room temperature, he kept this at 37 degrees, and he kept this in boiling water for a period of 1 year. Okay, did nothing, just kept it at these temperatures and saw what happens. What does one expect? I can't hear.
Equilibrium at some point.
Equilibrium at some point. Everybody okay with his answer? That is what one would expect. In reality, what happened was there was no reaction. There was zero product form. Okay, in all these cases. So people were confused, why is there no reaction? There's absolutely no reaction even if I keep it in boiling water for 1 year. So how does one study such reactions? Okay, remember you have to study this in the context of biology, so you can't use organic solvents, you can't use different things. So what they did is they took this same reaction and they performed it at very high temperatures in gas phase. Okay, and what they found was they started seeing a little bit of turnover, which is formation of product at very high temperatures, over 200 degrees. And based on that, you can sort of back calculate, so you can get rate constants and you can plot this as a function of temperature, and they found that at very high temperatures they were seeing some sort of reactions. And when they sort of backtrace this to get things happening at maybe 37 degrees or 25 degrees or room temperature, they got a rate. The rate of the uncatalyzed reaction, or K uncat, turned out to be something on the order of 2.8 times 10 to the minus 16th per second. Okay? Yes?
There is no enzyme in gas phase.
There's no enzyme in gas phase. There's no enzyme yet. Everything is uncatalyzed. So that's how they got the K uncat. Okay?
Only substrate and nothing else.
That's it. I'm just looking for this decarboxylation reaction in the absence of an enzyme, just in aqueous solutions. Okay? Now, as we heard from the last talk, the age of Earth, from what I googled, is something on the order of 2 billion years. Okay, 4.5 billion years. Turns out that if you look at the half-life of this reaction, it is approximately 78 million years. All right. Now you can see why DNA was such a stable molecule to life, for life to originate, because it's extremely stable by itself. Okay. Now, if we were to rely on this reaction to let life evolve, we would never evolve. So we evolved enzymes to facilitate the same reaction. Now, if you do the same reaction at room temperature in the presence of an enzyme, you get a kcat value, which is approximately 40 turnovers per second. So if you look at a very sort of simple reaction, you find that the rate enhancement of an enzyme over an uncatalyzed reaction is somewhere on the order of 10 to the 17th. Okay? So this is one extreme example to show you that enzymes catalyze reactions in biological settings at room temperature, which are otherwise almost unfavorable. And that's why you need enzymes in your body to function. Okay? Number one. Now let's look at one more example on the other end of the spectrum. The other example that I have for y'all is basically making soda. I am taking carbon dioxide and I am going to add it to water, and I'm trying to make carbonic acid. Heard of this reaction before? Yes. Okay. Now the second reaction that I have is I'm taking CO2 and I am bubbling this in water. Okay? And I am going to make basically bicarbonate. All right? This is principle of making soda also. Now, why is this reaction important? Why is this reaction important to everybody in this room? Yes?
CO2 dissolves in blood, so that
You want CO2 to be excreted as a gas, not get dissolved in blood. But you need it to be dissolved in blood for another reason.
Lungs would, the CO2 released from cells need to be transported to lungs to be removed from our body. So this is how it flows in the bloodstream.
Partially correct, but the main reason, yes, correct. So this is the reaction that buffers your blood. it keeps your blood at pH 7.4 to 7.5, anywhere between 7.2 to 7.5. So this is the reaction that creates the buffer in your blood. Okay, so biologically this is a very, very important reaction. So if you look at this reaction, the K uncat of this reaction is something on the order of 0.15 per second. So as compared to the last example, this is a fairly fast reaction. Okay, it has a T half of approximately 5 seconds. So fairly fast, okay, as compared to the last reaction. But this is very slow for biology, because if there is even this much fluctuation in the buffer of your blood, you can end up with conditions such as acidosis, ketosis, etc., etc. So that is why you have a dedicated enzyme in your body to keep regenerating the buffer. Okay, and the enzyme is an enzyme called carbonic anhydrase. All right. Now, the K-cat of this enzyme is approximately 10 million per second. Okay? The only thing that limits the K-cat for this enzyme is diffusion. Right? So you can now see that this enzyme has to be really, really fast, and it's a very abundant enzyme in your body to maintain this pH. But in terms of rate enhancements, it is only about 10 to the 7th. So it's not that much enhancing the rate, because the intrinsic rates of the reaction are already very fast. So you can see in the two examples that I've shown you that enzymes can enhance rates of very slow reactions very versatilely, or it can enhance rates of fast reactions by making them go faster. So typically in enzyme catalyzed reactions, you will find rate enhancements anywhere from 10 to the 7th to something like 10 to the 20th. Okay, so these are just two examples of how enzymes are enhancing rates. Now the next set of things I would like to talk to you about are factors that affect enzyme catalyzed reactions, and we look at three. We look at the effect of temperature, we look at the effect of pH, and we look at the effect of substrate concentrations on an enzyme catalyzed reaction. Okay, let's look at these three. So what does one expect with temperature? So if I do a very simple experiment where I am looking at rate of an enzyme as a function of temperature, in most cases I would find the following profile. So I'm plotting the rate of a reaction and I have temperature here. And what one typically sees is at low temperatures I have low rates. As temperature increases, my rate increases. I'll probably have a region where I have constant rate and then my rate falls. So this is, remember in biology, there are always exceptions to the rule. There is no theorem in biology. Okay, there are always very, very good exceptions to every rule. And we'll come to exceptions as we see things, all right? So why does this happen? Any guesses? So let's see what happens at high temperatures first. What happens at high temperatures? Denaturation. It's just like cooking an egg. So when you increase temperatures, you have protein structures which are destroyed, and therefore you have no reactions. This catalyst cannot function. What happens at low temperatures? At low temperatures, you have poor diffusion, and therefore you don't have enough constructive collisions between the enzyme and the substrate. and therefore your rate drops. So at lower temperatures, you are sort of limited by diffusion. At high temperatures, you're destroying the catalyst. And therefore, you need a certain sort of region where enzymes function best. Exceptions to the rule, if you look at, have you heard of polymerase chain reaction, PCR? Yes. The enzyme, so PCR reactions are done at 95 degrees. How can they be done at 95 degrees with an enzyme? Because the enzyme that is used in PCRs comes from an organism which lives in boiling hot water, Thermus aquaticus. Hence, the polymerase is called Taq polymerase. It comes from the name of the organism from which it was isolated. And this organism lives at 95 degrees, so it can function at 95 degrees. And therefore, all PCR reactions happen at that temperature. So the enzyme is stable at that temperature, therefore these reactions can happen at those temperatures. Okay, so that's the exception. Fine? Any questions? All right, now let's quickly look at the effect of pH. Now pH, before I get into pH, let's quickly look at amino acids and why amino acids play an important role in pH. Enzymes, as I said, are proteins. Proteins are polymers of amino acids. All right? Amino acids typically have a carbon, which is the C alpha carbon. It has a hydrogen, it has a carboxylate end, it has NH3, and it has R. Okay? This R is called the side chain of an amino acid, or the side group of an amino acid. And this side group has implications in the effect that pH has on enzyme catalyzed reactions. The N and C basically are responsible for polymerization, where you lose water between them and you form a peptide bond. Okay, so let's look at the different side chains first. Now side chains are broadly classified into three groups. You have acidic side chains, and these are nothing but groups having carboxylates, amino acids such as aspartates or glutamate, and these in all biological systems are deprotonated. Okay, so they have pKa's ranging from maybe 4 to 6, so fairly acidic groups. All right. The second group are basic amino acids, and these can be groups that have free amino groups on the side chain, such as lysine, which is positively charged physiologically. You can have arginine, which has a guanidino group, and again this can be charged positively under physiological settings. And these typically have pKa's that are 12 to 14ish. So this can abstract protons, this can donate protons biologically. You also have another group which are called amino acids with heteroatoms. Heteroatoms, typically side chains biologically will have carbons or nitrogens as their reactive groups, but you have heteroatoms which are molecules like cysteines, which have a thiol, or you can have serines which have hydroxides, and all of them typically have pKa's that range from maybe 8 to 10. So you can see that nature has put specific chemical groups with an array of pKa's to catalyze different reactions. And all these groups are involved in a type of catalysis which is called acid-base catalysis. Okay, so they basically take protons, donate protons, take protons, donate protons. And as a result, the protonation of these groups has tremendous effects on rates of biological reactions. Okay, now we'll see 3 scenarios of such reactions. Now, if I need a group, say I have an acidic group that is responsible for catalysis, okay? What one sees is a profile that looks the following. All right, so you have, I'm looking again at rates of reactions as a function of pH. Now, if I have a group which is acidic in nature that needs to be deprotonated for optimal activity, what one sees is that at higher pHs, the rate is constant. But as I go low in pHs, this group is going to be protonated, and therefore I will start losing activity. Make sense? Conversely, if I'm looking at a group where a basic residue is essential to be protonated for activity, what one finds is at lower pHs, this is going to be active, and as I go higher in pHs, I lose activity. In reality, for acid-base catalysis, typically you need a combination of these groups. And what one sees for a lot of cases is a profile that looks like that, where one group needs to be protonated and another group needs to be deprotonated for optimal activity. So if you look at different literature, you will find that these curves are called bell-shaped curves. And depending on the type of group one sees, you can classify it as a. So this is, for example, called a HA bell curve because an acidic group is involved. This can be called a HB bell curve because a basic group is involved. And this, for whatever reason I don't understand, is called a bell curve in literature. Okay, so you see such effects because of the protonation of side chains that are present in enzyme active sites. Now there are again exceptions to all of this. There are certain enzymes that function only at low pHs and certain enzymes that function only at high pHs. And they sort of disobey this rule. Examples of this, all the enzymes in your stomach, digestive enzymes in your stomach, they function at pH 2. Example of this is pepsin, which is a digestive enzyme in your stomach. It's only active at pH 2. Another enzyme that biologists routinely use for a lot of reporter assays is an enzyme called alkaline phosphatase. And this enzyme functions only at very high pHs. So again, this sort of obeys the rule of pH. Okay? Clear? Yes, no? All right, now we'll look at one last instant of where substrate concentrations affect rates of reactions. All right, so when one does an experiment, I'm taking one substrate and I'm getting one product as a function of enzyme. And when you plot a graph where one sees, where I'm plotting basically rate of the reaction as a function of substrate concentration, one sees the following. Okay, initially the reaction is, the rate is linear to the substrate concentration. All right, at very high substrate concentrations, I find that the rate is constant, and in certain region I find that the rate is variable. Now remember, these are people who are studying this from biology, and they want to develop a mathematical solution to a biological problem. Sometimes these can be mutually independent sets. So, they had to come up with very sort of simple explanations to why this is happening. So I'm going to break this graph into 3 portions. Okay, there is a region where there is a constant rate, where there is a variable slope, and where I am seeing a linear sort of relation. Okay, now there were 2 people in the early 1900s, a PhD student and advisor, who tried to solve this problem. Their names are Michaelis and Menten, and therefore the equation that is associated with this solution is called the Michaelis-Menten equation in enzyme kinetics. All right? Now, as I told you, we see the following in an enzyme catalyzed reaction. We see various steps. So what Michaelis and Menten did is they basically simplified this equation a little bit. The first thing they did is they made some approximations. What they said is, we know now that a lot of reactions in biology, chemistry is the rate limiting step. So we will, beyond this point, recommend that everything that happens after this is irreversible. So this equation now becomes the following. Okay, and they defined certain rate constants. They called this step as K2, which we now know is Kcat, or the enzyme catalyzed reaction. You have a K1 and a K1 inverse, which is the binding and the dissociation of the substrate to form and break the enzyme substrate complex. Okay, so that was the first approximation that they made. Okay, and now they wanted to calculate the rate of formation of the product, which is a simple sort of rate equation, which is K2 times the concentration of the enzyme substrate complex. Everybody able to understand this? Students? Yes, no? Okay. So now, remember, ES is an entity that one cannot measure. It's a very transient sort of complex, which is very difficult to measure, remember, especially in the early 1900s. So the only thing one can measure in this equation is the following. You can either measure concentration of substrate, you can either measure concentration of product, or I can measure the concentration of the enzyme that I've added in the assay. Okay, 3 things I can measure. So what people began to do then is they began to figure out how to solve ES, and they made another approximation. They said the formation of ES, because it's so transient, is equivalent to the dissociation of ES. Okay, so rate of formation is equal to the dissociation of ES. And this approximation in enzyme kinetics is called the steady state approximation. And therefore the Michaelis-Menten equation today is also known as the steady state enzyme kinetics equation. Okay, and if you solve for this, one finds that the rate of formation of is K1 times enzyme into substrate. The rate of dissociation is K inverse times ES plus K2 times ES. And so if all the arithmetic is done okay, you get ES, which is equal to sorry. So you get E times S, which is okay, and what this term is known today as is another term in enzymology, which is called Km, or the Michaelis constant, and we'll see what Km means in a little bit. So now if I basically take all these terms and I plug them back into the rate equation, okay, just to cut a long story short, I find the following. I find that the rate of the reaction is nothing but K2 times the total enzyme times the substrate over the substrate concentration plus Km. Okay, so basically this equation is known as the Michaelis-Menten equation today. Fine. Now if you take substrate concentrations at infinity, okay, what this equation simplifies to is nothing but K2 times ET. So you don't have any effect of substrate, and therefore at very high substrate concentrations the rate becomes constant. If you take substrate concentrations which look closer to 0, the denominator is basically much lower than Km, and therefore this equation becomes linear as a concentration of substrate, and between the two it becomes variable. Okay, now Km in enzyme kinetics is the concentration of substrate at which the rate is half the maximal rate. So if you now equate substrate concentration and Km, you will find that the rate is basically half the maximal rate. Okay, and Km basically defines how good a substrate binds to the enzyme. So the lower the value of Km, the better the substrate binds to the enzyme. The higher the value of Kcat, the more efficient the enzyme is in performing the biological reaction. So whenever you have an enzyme substrate pair, you have two fundamental rate constants. One of them is Kcat and the other is Km. And between these two, you can understand how well an enzyme and substrate pair work. Okay, so hopefully I have shown you today that enzymes are interesting. Enzymes catalyze reactions which otherwise would not happen in biological timescales. Enzymes are catalysts that function at optimal temperatures, pHs, substrate concentrations, and by doing so, are facilitating life. Now, what are sort of the next frontiers in enzyme or enzyme biology is something I will sort of prospect on in the next couple of minutes. Number one is, if you look at humans, okay, we have approximately 20,000 protein coding genes. Okay, they code for proteins, out of which about 10,000 or so are enzymes. We barely know the functions of 2,000 enzymes. So about 80% of the enzymes, we still don't know what they're doing in the context of physiology, in the context of real biology. So there is a huge effort in understanding functions of enzymes. Number two, people are trying to evolve enzymes. Enzymes are catalysts. They catalyze specific biological reactions. So people are evolving enzymes to catalyze non-biological reactions. For example, degradation of plastics. Catalyzing, with green chemistry coming about, with carbon footprints being discussed, enzymes are now being tailored to catalyze chemical reactions which are happening in industries. So that field of biology is called protein evolution. A third thing that's happening today is enzymes are being used as enzyme inhibitors are classical inhibitors for most drugs in the market, But now enzymes such as CRISPR-Cas9, they're being used to perform gene editing and change genomes of people to come up with therapy. So such sort of things are happening with enzymes. Okay. With that, I will sort of close. I will again congratulate everybody who's won the medal this year in the Olympiad. I'm sure everybody has. Also for all the NIIS students, I was once an NIIS student myself long time ago. So glad to see all of you all in the room as well. Thank you very much.
Thank you, Professor Kamat. Questions? Yes.
How does carbonic anhydrase capture CO2? Because carbon dioxide doesn't really dissolve in water.
Two ways. It uses whatever, so again, the dissolving of carbon dioxide in water is an equilibrium reaction. So there is some degree of carbon dioxide dissolved in water. As I said, enzymes like carbonic anhydrase are very, very abundant. They're present in millimolar concentrations in cells. So whatever carbon dioxide is dissolved, they're pushing the equilibrium towards making bicarbonate, so more carbon dioxide gets dissolved. So that's how it does the reaction. It also has a zinc in the active site that sort of polarizes the carbon dioxide.
Okay, there's a hand from here.
Sir, is there any relation between K1 and K-1? Like K1 should always be greater than K-1, right?
Depends on how, it depends on a few factors. It depends on, number one, how rigid the enzyme active site is, because if a molecule cannot get into the active site, it will dissociate. Number one. Number two is it depends on the size of the substrate. If the substrate sometimes is too large, for it to bind in the correct conformation is also not very productive all the time. So K1 and K-1 depend on the enzyme substrate pair. Not every molecule that binds on the enzyme is converted to product.
That's okay, sir, but if the enzyme substrate complex is also going further, right? So according
it has two options. It has an option of going back to enzyme plus substrate, or it has an option of going to enzyme plus product. And that depends on the enzyme and substrate pair.
So a certain fraction is going forward, right?
Correct.
So according to Lee-Chatelier's principle, because the product is getting removed, more and more product should be formed. So the rate of forward reaction should be more.
So the rate of forward reaction can only be more if there is free enzyme available. If there is enzyme substrate complex formed and that is stable and not going to product, there's going to be lesser product formed. So you need free enzyme also to be present. So it depends on a combination of Km, which is the 3 rate constants, and Kcat.
Okay, sir. Thank you, sir.
You mentioned you work on enzymes and you use engineering, biology, and chemistry. I understand biology and chemistry. Where does engineering come into this?
Engineering comes because we work a lot with lipids. Okay, so we work on enzymes that make or break lipids. And turns out that if you look at lipids by themselves, they have no chromophores whatsoever. They are boring aliphatic carbon containing compounds. So what we do in terms of technology is we do a lot of mass spec, which is a technology to measure masses of molecules, mass spectrometry. And we're developing technologies to measure lipids, both in free systems, in cells, in tissues. So that is where the technology development comes. I didn't talk about it.
Thank you.
Okay.
Sir, can you hear me?
Yes.
Yeah. While drawing the graph of the
You still have to speak a little louder.
While drawing the graph of the reaction for the enzyme catalyst, so you first drew for the same reaction of first the substrate and then the enzyme substrate complex, and then the enzyme product complex, and then enzyme and product. So you drew enzyme substrate and then there was an activation after which the enzyme product was formed. Can you please explain why there should be an activation there also?
That is the chemistry. So I am doing a chemical transformation. It can be a simple thing like removing a proton from one place, putting it on another, or in the first example, taking out a carbon dioxide. And the chemistry, in most cases, is the rate limiting step. Therefore, it's going to have the highest energy of activation barrier to cross. Because once I cross that barrier, everything seems to be energetically downhill.
But the thing is, it is still in the enzyme, right?
It is still in the enzyme, but because I've crossed the barrier, steps downstream become faster. So remember, enzyme is a dynamic structure in solution. It's not a rigid thing. So the moment I have the formation of a product in the active site, the conformation of the enzyme is going to change to facilitate the dissociation of the product. So it's a very dynamic process of catalysis.
Thank you.
Yeah, please.
Hello, sir. So my question is, you mentioned about green chemistry. So I would like to know about how green chemistry is utilized, like atom economy and catalysis. The principles are utilized in the
how it is used. Okay, I'll give you an example. So basically, this was Nobel Prize winning work from Francis Arnold's lab about 3 years ago or so. So what they're doing, for example, nitrosylation reactions don't happen in biology. But they're taking enzymes to do these reactions. They're making enzymes do carbon-carbon coupling. They're making enzymes do radical reactions. So they're making them do a whole bunch of things based on the principles of catalysis. Now, all the other catalysis that are used are metals, or they are very toxic compounds which have high carbon footprint. So all governments in the world want to reduce CO2 emissions. They want to make processes more sustainable. That's how this field is emerging.
Thank you, sir.
So can you just tell more about your specific field of interest?
Just a minute. Please take the mic.
So can you
Yeah.
So can you just please tell more about the specific field of interest of your
For my group?
research? Yes, sir, of yours.
So my group is interested in a class of lipids which is called bioactive lipids. All of us have heard about triglycerides, cholesterols when one does blood profiling. But there are very poorly abundant lipids in your body which act almost like hormones. Okay, and they control functioning of your brain, your immune system, your cardiovascular system, etc., etc. So what my lab specifically does is two things. One is we are trying to figure out what are the enzymes that either make or break these lipids. And we're trying to develop technologies around identifying newer such lipids in biology and figuring out what role they have in physiology. That's the broad aim of my lab.
Yeah.
Sir, I wanted to know about the evolution of enzymes, like how enzymes are proteins. So what are the prerequisites, or how do the transformation occurs?
So how proteins evolve during evolution, basically?
No,
how protein...
how do how do enzymes form, and how can we further from the evolution process? I want to
So
know.
from an early enzyme where we are now is what you want to know. So as I said, enzymes are catalysts. Okay. So if you look at enzyme structures overall, you will find that there are maybe 3 or 4 generic type of structures. Okay. And what has changed over time to accommodate, so 4 generic structures catalyze in excess of 50,000 reactions. How do we do that? Is simply by changing the permutations and combinations of what is present in the active site. So the residues that line the active site or residues that bind substrates change as a function of evolution. So you will find that an ancestral bacterial enzyme and a human enzyme, if you sort of overlay them, they look very similar overall. But they do completely different things because there are maybe 10 or 20 amino acids that have changed as a function of evolution. And that has enabled them to do completely different things. So that is the same principle people are using to evolve enzymes to do non-biological activities. You keep the overall structure the same, but you're tinkering with residues in the active site to accommodate different reactions.
Okay. And so, like in the process of natural selection, like did it occur naturally, like those permutation combinations?
Your guess is as good as mine. I would say natural selection, yes, but
like, sir, right now we talked about enzymes speeding up reactions. So are there also enzymes used in biological processes to slow down reactions? Is that helpful?
so biology will not invest in something which is going to be counterproductive. So it will not do the reaction at all if it needs to slow it down. It will find mechanisms to slow it down another way. For example, it will, if you have compounds, so for example, for us, when you eat glucose, not all of it is converted to energy. What we do is we store glucose as glycogen and use it when you need it. Okay? You don't need to evolve enzymes to slow down the process of burning glucose for energy. You'll find other mechanisms by which you will slow down biological reactions.
Thank you, sir.
Any more questions? Okay, if not, let's thank Professor Kamat once more. And so that brings us to the end of the morning session, and we'll take roughly a half an hour break and come back here at 12 o'clock for the main function. There's tea outside, just outside, so I request all of you to please go out and have tea and come back at 12 for the award.